In SARS-CoV-2 antigen rapid test kit, we provide nasal swabs, 1 bottle of sample treatment solution, and a test strip. The operation process in this video is different from the one shown on the strip. Please follow this video. First, you should use the nasal swab to collect the sample. Carefully insert the swab into the nostril and using gentle rotation to push the swab until resistance is met at the level of the turbinate, which is less than 1 inch into the nostril. Rotate the swab 5 times or more against the nostril wall. Then slowly remove from the nostril. Using the same swab, repeat sample collection in the other nostril. Thread the swab head through the bottom of valve B into valve A at a 45-degree angle to the strip. And then drip 6 drops of the sample treatment solution on the swab head. Rotate the swab 2 rounds clockwise and counterclockwise. Remove the protective cover of the fixing glue. And close the strip. Press lightly to make sure the strip sticks well. And start timing. After about 15 minutes, you can read the result in the window.